Hello, everyone. This is Pocket Size Bio with Dr. Casco, and today we're going to talk about coding and non-coding DNA. Right? And before we get into this particular topic, I just want to go over a little bit of review on what DNA is. So let's get to it. So DNA, as you know, is the blueprint of life, right? Contains all the information needed for an organism to grow, develop, thrive in its environment, reproduce, and pass on within a species and a population. The genes, right? Basically all about heredity. But in general, when you want to focus on these kind of topics, you need to talk about DNA, right? And how DNA works. And if we look into the building blocks of DNA, you know that DNA is composed of 4 nucleotides, now DNA only, right? Adenine, thymine, guanine, and cytosine, right? Designated A, T, G, and C. And the sequence in which these nucleotides are composed or are in an order, right, will define a particular information, right, that will be coded into the sequence, right? And essentially that's what genes are, right? They're particular segments of DNA in which the nucleotides are in a particular order to code for something, for some protein, right? So these bases repeat in a particular order or sequence that encode information to create a polypeptide, right? We know that polypeptides are basically chains of amino acids, basically the building blocks of proteins, right? To create these molecules that are essential for life, right? Here in these diagrams, you can see basically how you have the double helix of DNA, which is composed of 2 strands of nucleotide sequences, right, that pair against each other, right? And you only have these 4 that are repeating in certain orders, right? If you look into this DNA molecule and you zoom in, right, to see each individual nucleotide, you see that each nucleotide, right, having that sugar in the phosphate, but that nitrogenous base is what hydrogen bonds to its complement, right? C with G, A with T. In essence, this is what DNA looks like. This is the general structure of DNA, okay? Now, when we go more specifically on what type of information DNA has within an organism's genome, right, there starts to become little subtleties that we need to take in consideration, right? Certain areas of DNA have the ability to code or have the potential to code for a particular protein, right? You refer to those areas of DNA as coding DNA, what we commonly think of as genes, right? So here you have this diagram where you have this cell and you have this nucleus, and inside you have the DNA, and you zoom into that particular section of DNA, even within the strand of DNA, not the entire strand of DNA, right, will code for something, only certain sections of DNA, right? So let's say here you have gene 2, and this broadly kind of will code for gene number 2, whatever X gene it is, right, related. And gene number 1 is here in another region of the whole genome, right? But even within little sections of that gene, you have some spaces that you really don't use when you produce a protein, right? But I'm getting a little bit ahead of myself. For now, I want you to understand that there's certain regions that DNA has that codes information to make a protein, and there's some spaces in between certain genes that there's no coding um, um, for a protein, right? Doesn't mean it's not useful, but it might have certain other functions, right? So a large portion of DNA doesn't code for a protein, and that is called non-coding DNA. Okay. And then as we understand how there's certain coding areas of DNA, how that actually passes on to create a protein is through a process called transcription and translation, right? And DNA basically serves as a template to create an intermediate molecule called RNA that we're going to get into in the next video, right? And then the information carried on from that RNA basically will produce a protein, more specifically, an amino acid sequence that will serve as the basis to produce a protein, right? Now, scientists have figured out what particular sequence codes for a particular amino acid. And we know that nucleotide sequences work in sets of threes, right? In the case of within the RNA landscape, it's referred to as a codon. Okay. More on that in the following video. But scientists have figured out that each codon, right, each set of three nucleotides for that particular codon codes for a particular amino acid. Here you can see this little table here that we'll get into in a lot more detail. I'm just showing it here for you. It's often referred to as the genetic code, right? And it's basically how the nucleotides are in a particular sequence to code for a particular amino acid, right? And we use this at the mRNA level, right? Because the mRNA sequence is what's going to be used by the ribosomes basically to start manufacturing that amino acid sequence. Okay, and that physical process is essentially protein synthesis. Okay. So this whole idea of gene expression and protein synthesis basically is broken down into transcription and translation. During transcription, you're basically producing an mRNA molecule from a DNA template, and then translation, you're basically using that mRNA that was produced, reading it to produce an amino acid sequence, right? Ultimately to produce a polypeptide, which can further produce a full protein. Okay. Transcription occurs in the nucleus, and translation occurs in the cytoplasm for eukaryotic cells. Okay, that's it for this video, guys. In the next video, I'll talk about transcription in more detail.